Hello everyone and welcome to our NGS-CN Summer School online collection on spatial transcriptomics. Before we jump to our next speaker, let's take a look once more who we are. So it's a next-generation sequencing competence network that consists of 4 production sites, as you see on the map. So in 2019, DFG has funded centralized sequencing applications. There were quite a lot of calls, and within each call there were a lot of projects. And from 2024, DFG has changed the strategy towards individual sequencing applications. That means that now there is no special deadline for a centralized call, and you can co-apply at the DFG together with the chosen genomic facility. If you wish, here is a QR code, contact us, and then we could take a look and maybe sequence together. If you want to know something more about our news and upcoming events, then you also can take a look at the QR code, scan it, and get into our mailing list. Let's grow community together. And with that to be said, let's welcome our next speaker.
Now we're going to go from what we learned yesterday from single cell. We'll bring that into spatial context. So we're going to first have a look at the spatial transcriptomics technologies that are out there. So to recap a bit from yesterday, first we said yesterday we had bulk RNA-seq, and with this is like you're making a smoothie out of your tissue, right? And then we had single cell sequencing, and we could start now detecting our individual fruits. But now we want to see, we have a spatial, and now we can see the whole cake. And this is important, of course, because there are many diseases or developmental processes where the role of a cell is also affected by the environment where it is. So let's have a look at another example of where a spatial would be useful. Suppose that you're studying, for example, brain development, or you're studying brain disease, and you want to see how these processes affect the structure of the brain. In this case, one way that you can go is take your brain, make sections, and then do some detection of individual genes. So for example, and in here you can see some in situ hybridization, so you could detect 1, 2 to 3, 4 genes per slide. This is a very valuable resource, but of course it has a low throughput. If you would like to see hundreds of genes at once, it might not be the best way to go. So in that case, if you need a higher throughput, what we can do is take a part of our brain, extract the RNA, and go for RNA-seq. In that case, then we would find our, for example, differentially expressed genes, and we can then start from there in a higher throughput. Another thing that we can also do is make a single cell suspension and go for single cell RNA-seq. And in this case, we can identify our clusters and cell types. But still, after going through these methods, it's usually useful to go back to the tissue and see where do I have these differentially expressed genes, or where is this new cluster that I found or that is affected in our tissue. And this is when spatial transcriptomics comes very useful. So what is spatial transcriptomics? This is a group of methodologies that allow the quantification of mRNA expression of a large number of genes, at the same time preserving the tissue context of the gene expression. So what we get from these spatial transcriptomics technologies is first, we retain the histology information. For example, here we can see it in a DAPI image. And at the same time, we can see in the tissue our cells, our clusters that we identify with the transcriptomics. And we can also go, ideally, to each individual cell and see the level of expression of hundreds or thousands of genes at once. How do we technically manage to do this? So there are 2 main groups of spatial transcriptomics technologies. One are called sequencing-based technologies, and the other one is the group of imaging-based technologies. What this means is that for sequencing-based technologies, basically for the readout, you need a sequencer, and for imaging-based technologies, you need a microscope. Now, in general, these 2 groups have quite different workflows, so I will walk you through first through sequencing-based technologies, and then we will go to imaging-based technologies. The general concept of sequencing-based technologies is that we have barcoded surface. So we have a surface with different capture oligos, that we call them, and each of these capture oligos has a different barcode according to the location, to XY location in the surface. These oligos, then we put our tissue of interest on top of these oligos of the surface, and we permeabilize the tissue. Before permeabilization, of course, we can, in some of the technologies, we can take an image of our tissue to retain the histology information. And then with permeabilization, release the mRNA from the tissue, which is then captured by our capture oligos. So how does this work? It's actually very similar to what you have seen yesterday from single-cell transcriptomics. You have captured oligos with a poly-DT that will capture the mRNA by the poly-A. And of course, each of these has a spatial barcode that will give us then the location information. So after capture, we do a first-strand cDNA synthesis, where we obtain our first-strand cDNA with the transcript information and the barcode information. So after we have a first-strand synthesis, now this first strand is actually covalently linked to our surface. So to be able to work with this, we need to do a second-strand synthesis to then denaturate it from our surface and then put it in a tube. And then we can continue in the same way as was explained yesterday for single cell with library prep and sequencing. And you can do short-read sequencing or long-read sequencing according to which information you're interested in. How do we now get back this transcript information into our tissue information? We have, as I said, we have a barcoded surface, and we know the XY location of each of our barcodes. At the same time, we take an image of our tissue, and we use these called fiducials, which are physical marks on the slides that then allow us to overlap these two sets of information. And then we can know where all these barcodes are located in the tissue. In the read one, so we have first sequencing the spatial barcodes, and this will tell us where in the surface the transcript was coming from, which then we know where in the tissue the transcript was coming from. Then in the second step of sequencing, we have the transcript information. And now we know that this transcript that we are reading here is coming from this location in the tissue. What do we get after this? What is the data we get after this running in a spatial transcriptomics? So we get our H&E image, when the known locations of the barcodes. And as Andreas explained yesterday, after processing the sequencing results, we get bind files. And with this, we can generate now. So yesterday we were talking about cell by gene matrix. Today we have to talk about spot by gene matrix, because the information now we get per spot. Sometimes these spots will match with the cell, single cell, and sometimes they don't. But we can still work with this spots by features table. Many times we work with these tables just very similarly to what we do with single cell results. But of course, there are more advanced methods where we can also, and importantly, introduce the spatial information. And this, how we analyze this, we'll see more in detail in the following talks. Now I will show you a bit the different sequencing-based technologies that are out there. And they differentiate mainly on the way they barcode the surfaces and in the resolution. One of the more widely used technologies is from the NGS Genomics. It's called Visium. In this case, we have, as I said, a barcoded surface. And in this version, we have spots with a 55 micrometer diameter. This means that each of these spots will not have a single cell, but probably around, depending on the tissue, 10 to 20 cells. And we also have an empty space in between. So the resolution you can see here is 55 or 100 micrometers. Then later, Visium HT was released. And in this case, the resolution increased massively. Now what they provide is a surface completely covered with barcodes. And each barcode has its individual barcode occupies an area of 2 micrometers by 2 micrometers. But now when you start increasing your resolution, you start going into issues of the mRNA capture efficiency. So in this case, then the resolution is not really 2 micrometers, but probably you need to be in some of these spots to get meaningful data to work with. Another technology that is available is CurioSeeker. In this case, it's not the oligos that are printed in a surface, but are bits, individual bits that have the barcodes, the oligos with the spatial barcodes. Each of these bits has the 10 micrometer diameters, which means that it's closer to a single cell resolution. And luckily, we're not able to test it as it's not available yet in Germany. But another issue that to keep in mind in this technology is that we cannot really take an image of the tissue before we start the process, but still the data is very useful, and according to the papers, it's looking anyway very nicely. Then Curio released another method. They managed to get a real single cell or single nuclei resolution. Similarly to the one above, then we have a surface barcoded with its 10 micrometer diameter bits. But in this case, after placing the tissue on top, there is a UV light that will release the barcodes, and these barcodes are incorporated into the nuclei of each of these cells, of the cells on top. The next step is to make from this tissue a single nuclei suspension and go for single cell or single nuclear technologies. Of course, it has a benefit that it's really a single cell. And another benefit is that when you go for single cell technologies, then you can go, for example, for multi-omics or attack. So you can really have spatial attack-seq, for example. So the drawback here is that the amount of cells that you need to sequence is quite high, and you lose quite some cells, so it becomes a bit limiting on the costs. Another two methods that are available for bits, so for surfaces coded with bits, are this one that I cannot pronounce the name, sorry. In this case, the bits have 2.5 micrometers diameter, so it really increases the resolution. But as I said before, there is a drawback on the efficiency of the mRNA capture. So even though the bits, the barcodes have a higher resolution, you need to be in many of these barcodes to get meaningful data. And then the last one with the bits surface is this called high definition spatial transcriptomics, where we have 2 micrometers bits. Again, we have issues with the low capture mRNA. Another method to barcode the surface is with microfluidics. In this case, we don't really barcode the surface, but we put the tissue on our storage slide, and then we deliver the barcodes with microfluidics. The benefit is that we can place known barcodes in it. First, there is a deposit in this horizontal direction. Each line will have a different barcode that will be attached in the cells through a poly-DT, so it will bind to the poly-A. Then there is a second delivery of barcodes in the perpendicular direction, and again we have a different barcode in each of the lines that will bind to the original barcode by ligation, and then with this combination, each of these spots will have a different barcode. And I think currently it's commercially available with the 25 micrometer resolution. Again, we have an issue with the low capture efficiency, but so the good benefit of this technology is that you can really combine with a lot of epigenomics readouts. So they have used it for different chip-seq detection and also for open chromatic with attack. And last, the last method to barcode surfaces is to repurpose sequencing flow cells. So that means we have a flow cell from, for example, here from Illumina. And what we do is to sequence a library of barcodes. And with this, we can barcode the flow cell. And the drawback is that you have to, of course, sequence this flow cell to get the barcode and the positions. And then you have to cut it and put the tissue on top, and then make your library again and sequence again. So it's kind of a bit laborious, but on the other side, it's cheaper. And you get a very good resolution. Of course, you have to be in, as you said, you will not get 0.6 micrometers. You have to be in a bit of data, but it's promising so far. All of these methods that I showed before require that you put a fresh tissue on top with a good RNA quality. But sometimes this is not available. Sometimes we have an FFPE block from the clinics, or when we try to make a fresh tissue and we section it and the quality is not looking good, like Anche explained yesterday, then we can go for other methods that are panel-based. In this case, we can use samples, for example, treated with formaldehyde, that the formaldehyde cleaves the mRNA, but since we are using proofs, so in this case, these are panel-based, we have proofs that have 2 parts, each of the proofs. And each one has around 25 base pair complementary sequence to our target mRNA. Then we incubate with these proofs overnight, and then we have a ligation step. And this ligation step increases the specificity as proofs that will bind unspecifically to the tissue, as usually happens, they will not be amplified. So then we will amplify with these known sequences outside at the later step, and those that were bound unspecifically and did not like it will not get amplified. So far, this is panel-based, so you have to design a panel. The drawback is it's so far only available for mouse and human. And all these panels include all protein coding genes. So you have to take into account if you are interested in, for example, non-coding RNAs might not be the way to go. And then the last benefit that this method has is that you don't need to work directly on the expensive barcoded surfaces. You can section your tissue on a normal histology slide. You can check that it has the section, the position, the quality that you're happy with. And then we have there is this machine called CytAssist that creates a sandwich between your tissue that has been incubated overnight with the proofs and the barcoded capture area. And with this, in this machine, there is a process of RNA degradation, degradation and tissue permeabilization. Now the proofs are the ones getting released and not the mRNA anymore because there is a RNase treatment. And then the proofs are captured in our barcoded surface. This is my overview of the sequencing-based technologies. There are even more out there, but I think enough with these technologies. As I said before, after you have run your sequencing-based experiment, you get this spot by picture matrix. And as was shown before, was shown yesterday by Catherine, then you can analyze it and get, for example, the nice U maps. And you can identify your clusters. And now the main difference is that you can identify directly all your clusters directly in your tissue. You can visualize, of course, individual clusters. And if you come from doing a lot of immunostainings or in situ hybridizations and you like to have a look at your data, you can, of course, then visualize each of your genes independently and have a look at all of them. Might be a bit too much, but it's another way to maybe at least get used to how the data looks like. So with that, I went through many sequencing-based technologies. And now I will switch to the other main group of spatial transcriptomics technologies, which are the ones that are imaging-based. So the imaging-based technologies are based on in situ hybridization. So they are basically a multiplex in situ hybridization. Now, when you do in situ hybridization in the lab, if you have done it, you can get maybe 1, 3, 4 genes in one tissue section. So how do we get from this to getting hundreds of thousands of genes in the same tissue? So there are different technologies out there as well for imaging-based technologies. And what they need to, the things that they have to achieve is to work on the limitations that you have in a normal in situ hybridization. For example, they have to find a way to overcome the limited number of colors that you have for imaging. They have to increase the signal intensity, the signal to noise ratio, and as well, they have to take into account the optical image intensity when you want to image hundreds of transcripts at once in the cell. So how they do this, it's different between the different technologies. And the main difference that they have is in this type of signal, how they increase the signal intensity and increase the signal to noise ratio. But there are many steps that are actually in common from these technologies. So I will go through first all the common parts. So as I said, these technologies are based on in situ hybridization. This means that we have probes that will hybridize to our targeted mRNA. But in all these cases, the probes will have two parts: a binding domain that will hybridize to our mRNA. So here, this little part here, or these parts here. And all these probes have as well a readout sequence. Now, the readout sequence is there because then we have a second round of hybridization with the readout probes. And these probes are usually fluorescently labeled. And then they will bind to the encoding probes. Now, after we have done the encoding probe hybridization and the readout probe hybridization, we will go for imaging. Now, if we only do this, we are back to the limitations of limited number of colors, right? The thing that all the technologies also incorporate is a way to remove these readout probes. So they incubate with one set of readout probes for a subset of the genes. You take an image, and then you remove these readout probes, and then you start again a second round of hybridization for another set of genes. Suppose that you have 2 colors, right? And then you want to image 100 genes. If you do this of detecting 2 genes per round, imaging and proof removal, then you have to go over 50 cycles to get to 100 genes. So what these technologies do, they use a different approach. Instead of an additive approach, the linear, they use a combinatorial approach. What this means is that each transcript is actually imaged many times over the different rounds of hybridization, imaging, and removal. And what gives us the identity of the transcript is not the image in one cycle, but the combination of colors over different rounds. So you can see here the combinatorial approach. So with this, with a few cycles, you can reach to hundreds of genes right away. This is theoretical. It's not really that we have here, like with 7 cycles, you can reach 100 genes, because of course you have to save some barcodes for negative controls. You have to design the barcodes in a way that if there is an error, you can still identify them. So the number of cycles is actually a bit higher, but of course much lower than the usual to the linear approach. Now, as I said, the way they amplify the signal will be different for each of the technologies. So I will explain this. I will go through these 3 technologies, but the same as sequencing based, there are even more out there and more coming. The field is growing very quickly right now. So I will start with Xenium. In the proofs for Xenium, the first round of proofs, they have a structure that is called padlock proofs. This means that they have RNA binding domains in the 5' and 3' extremes of the proofs. These 2 sequences, then they have to bind next to each other in the mRNA in the target site. If they bind next to each other, then there is a ligation step, and this will get ligated. And this step of ligation increases the specificity. So if the proofs will bind in the tissue somewhere, not specifically, then this ligation step will not occur, and then we will not detect these unspecifically bound proofs. After this, there is a round of rolling Silker amplification. And this is the main approach that this technology has to increase the intensity of the signal. Important thing that I missed to say, the proofs have as well a barcode in the backbone, right? This barcode is specific for each of the genes, and this barcode gets amplified during the rolling Silker amplification. And then we have our second round of hybridization with the fluorescent proofs that will bind to this amplified nanoball, we can call it. Yeah, we will have a higher intensity of fluorescence for the imaging. And as I explained before, it's not that each gene will be imaged at 1 by 1, but there is a combinatorial approach. And let me show you maybe this image that is nice to have it more visual. If you look, for example, at these spots here, you see that in the first cycle, this position is pink. The next one is blue. And in the next one is green again. And then after this, the color of the spots is recognized over the different cycles. This is compared with the barcodes for each of the genes, and then we can get the identity of the genes. Another technology that is out there is called Cosmics from Nanostring. And in this case, they have a different approach to amplifying the signal. So they have this tree-like structure. So we have here the first proof, specific proof that binds to your mRNA. It has a 35 to 50 base pairs, nucleotides that will bind to the mRNA, and also has 4 readout domains, where our readout proofs will then bind. And each readout proof then has this tree-like structure where all the readout proofs with fluorescent will bind, amplifying a lot the signal. And the way they manage, so they have first you hybridize, as in all the technologies, you hybridize with the proofs for all your genes. And then after imaging, you can remove the fluorescence of the readout proof by a photocleavage site. And then you go to the second round of hybridization, which will use the next readout domain, in this case. And the third technology is MERFISH. In this case, the way they have to kind of somehow amplify the signal is to have many proofs binding to the mRNA. So you have at least 50 encoding proofs, and then each of these proofs has as well the readout sequences that we can see here in more detail. So each of them will have 3 readout sequences where the fluorescent proofs will bind. And the method that they use in this case to remove the fluorescence in each round is by chemical cleavage of this bisulfide. So the fluorescent molecule is attached to the readout proof by a bisulfide bond that then can be cleaved after each round of imaging. Now, in this case, there is no really, it's a closer single molecule fish because there is no real an amplification of the proofs. Then they have to take a different approach to remove the background to increase the signal to noise ratio. And the way they do is, after hybridization with the encoding proofs, they go through steps of embedding and digestion. So they clear out the tissue to have only the signal from the encoding proofs during the imaging step. And this is how they have a much higher signal to noise ratio. Now, we went through the 3 different technologies. But another thing that you have to keep in mind is, of course, now we are talking about a targeted approach. So you have to design your proofs for the genes of interest. And usually you can design from hundreds to thousands. So when you're talking about thousands, it's not really like you can choose which genes you're there. You have panels that you can use, designed by the companies. But still, when you want to design your panel, one important thing that you have to keep in mind is the optical crowding. If you want to detect either too many genes in one cell, or you want to detect very highly expressed genes, then you will have to come to this situation where the fluorescence will just be too bright, they will touch each other, and you will not be able to distinguish individual spots. So the process of panel design is something that is a very important step for these technologies. There are softwares and the support from the different companies on how to do this, but it's something that you really need to take into account. So which kind of data we get now from the imaging-based technologies? In general, we get, for example, a DAPI image, and then we have transcript locations that we get from resolving all the individual spots from the imaging rounds. But to analyze it, usually sometimes it's useful to get these transcripts assigned to a cell type, to a cell in the tissue. And how do we assign then these transcripts to the cells? One way to do it is to apply some way of doing cell segmentation. I will explain you now, in this case, how this is done for Xenium, but this is similarly done. Analogous methods are also available for MERFISH and Cosmics. But what we have is, at the same time as we are doing the experiment, we have boundary stains. So we have different possibilities for for cell boundary stainings. And we have as well interior protein stainings, plus the DAPI. And for example, in the case of Xenium, what the software does is first detect the boundary staining, try to find a boundary with this information. If it does not find it, then it will use the interior stainings to delimit the cells. And if none of these have worked for a certain cell, what it will do is to take a nuclear segmentation and expand it, for example, 5 micrometers to define a cell. And then according to the limits of the segmented cells and the location in XYZ, we can get again our cell by gene table. So here we can see a bit more in detail. We have now the nuclear segmentation. Then we have cell segmentation, so visualize. And here we can see our transcripts on top, right? And then bioinformatically, all the transcripts here will be assigned to this cell. So after this, you will have cell segmentation information, like the location of the cells, how many counts you have per cell, the area, and in addition, as before, you again a cell by gene count matrix. This is to repeat, more or less repeating what I already said before. You can analyze this data, get an nice UMAP, and then find again your clusters in the tissue. And as before, you can also go crazy and look at hundreds of images individually if you like it. Of course, there are more advanced ways to analyze this and use the spatial information, which we'll go through later in the other talks. So to summarize, we have sequencing-based technologies, and here the resolution can vary a lot from 55 micrometers to 2 micrometers. The big advantage is in this case is that you can get the whole transcriptome because you capture the mRNA. And in the case of imaging-based technologies, the resolution is determined by the imaging that you do and some other aspects, and then you can reach to like even 0.2 micrometer resolution for each of the transcripts. In the technology, you can go for a few hundreds to thousands of genes, but of course you cannot go to whole transcriptome. Another benefit here, as I said, is that you can really go through thousands of whole transcriptome, but in this case you don't. But the benefit here is you can have a higher sensitivity. For example, if you're interested in a lowly expressed genes, then since you are targeting it, the sensitivity is higher than the whole transcriptome approaches for lowly expressed genes. In general, you can apply both technologies to all tissue types like fresh tissue, fixed frozen, FFP. How to choose this, as we went through the talk yesterday with Anche. But if you want to discuss more, of course, we can always talk later. You can contact us. Just the last note, keep an eye, because if you're interested in technologies, there are many more that will be coming soon. Here, ABITI and this G4, ABITI24 actually. So these are sequencing technologies. And they promise that you can put your tissue in the flow cell, put the tissue directly in the machine, and sequence the RNA directly in the tissue. This should be coming out actually the second half of this year, and I'm looking forward to see if this works. And there are also 3D spatial transcriptomics technologies that should be coming. With this, I would like to thank, and I would like to let you know that these technologies involve actually many different expertise, and it's not something that you would not do it on your own. And how we manage to make it work here is to collaborate with we collaborate with histology facility to process the samples, because as you heard from yesterday from Anche, to get this practice on sectioning, it can be quite nerve-wracking, but we already have the experts here. Then we collaborate with the microscopy facility for the imaging. Then we take care of the spatial workflows. It would be nice to have an image there, but next time. And of course, then we have to analyze this data. And for that, we work with the bioinformatics unit. And of course, the person with the biology question, of course.